Hello students, good morning. I'm your educator Dalsy, and today we are going to do our next topic, that is outbreeding. What is outbreeding first? If the flowers that are hermaphrodite means they are bisexual, means both the male and female parts, that is stamen and the stigma, are present on the same flower. So due to self-pollination or self-fertilization, these hermaphrodite flowers, they undergo inbreeding depression, in which they lose the fertility or the a kind of good growth due to inbreeding with the same kind of characters. So outbreeding occurs when there is appearance of inbreeding depression within the hermaphrodite type of flowers. So the hermaphrodite flowers, when they undergo inbreeding depression, it leads to the discouragement of self-pollination. With inbreeding depression, the chances of self-pollination reduces, and it encourages, means it increases the incidence of the cross pollination. Means inbreeding depression, it leads to the development of some kind of characters in the flower which ensure the cross pollination within flower. Cross pollination is the process in which the pollen grains of one flower or the pollen grains of a flower of one plant are transferred to the flower of some other plant, whether of different those flowers should be present on different plant. So to ensure cross breeding or cross fertilization or outbreeding, some kind of characters are observed in the plant, that is, if the pollen is released at a different time from the time of reception of stigma, it means pollens and the stigma, they mature at a different time. It means they lack synchronization. Synchronization is the process in which they develop at the same time. If they lost that property of development at the same time, so they are said to be the non-synchronized type. If the pollen matures first, then it will not be capable to add or stick at the stigma, so there will be no fertilization or no pistil pollen interaction within the both. Next, the anther and the stigma, they are present at the different position. They are present in such conditions that the pollen grains, when they shed off, they will not shed off on the stigma or the receptacle part of the female carpel. Next, they lost the compatibility, means they are self incompatible, means they are not capable of doing or performing self pollination. They lost the capability to fertilize their own stigma or their own egg. Last one is, ultimately all these characters result in production of the unisexual flowers, means male flowers are present at some other plant and the females are present at the other plant. So with the development of unisexual flowers, the chances of outbreeding and cross pollination increases. Next is the pollen pistil interactions. So what about this? The pollen means the pollen grains, pistil means the female. The interaction between the both are said to be the pollen pistil interactions. As you know, if you want to see the fertilization, the primary point there to notice is how the pollen grains are transferred to the stigma. So as you know, most number of pollen grains shed up on the stigma, but only one or two or few of those are capable of doing fertilization. Not all are capable of doing fertilization. So on that basis, pollen grains are divided into two types. They can be compatible, means they are capable to be accepted at the receptacle or the stigma part, or they can be incompatible, means they are not accepted by the pistil. If they are compatible, it means they are accepted. If they are incompatible, they will be rejected by the pistil or the stigma. So if they are rejected, there will be no pollen tube and no fertilization will take place. But if the pollen grain is compatible type of that of stigma, so it leads to development of the pollen tube, and that pollen tube ultimately crushes the food or the nourishing tissue present in the style. So it primarily releases the It primarily releases the type of hydrolytic type of enzymes which degrade the nourishment tissue present in the style region. So it is necessary for the development of the pollen tube, so that the pollen tube is capable to crush that style and reach up to the ovary. After reaching the ovary, that three-celled pollen tube, means one with vegetative and two with male and two male gametes, it enter at the ovule, through which end? Through the micropylar end of the ovule. It enter at the micropylar end. From that, around the egg, there is egg apparatus. Around the egg, two synergids are present. So the pollen tube enter the synergids through filiform apparatus, and those synergids of filiform apparatus, they guide the pollen tube, or they provide the path for the pollen tube, so that the male gamete get fertilized with the egg. So one of the male gamete, it mates with the egg, and it is said to be the syngamy, and it results in the formation of the zygote, that is the seed. And on the other hand, if the other male gamete, the second male gamete, it mates with the polar nuclei, that is 2n polar nuclei, then it undergoes the triple fusion, means it results in formation of 3n type of tissue, that is said to be the primary endosperm, which provide nourishment to the growing seed. Both these events collectively known as the double fertilization. So what is the benefit of all this? Via this we are capable of doing the plant breeding experiments, in which we ensure that which kind of flowers will do self pollination, and how can we ensure if they are showing self pollination, so how we can induce cross pollination in them. So necessary point here to notice is we are interested in deriving specific kind of characters in the plants which are not present, which are not found to be present in single species. They can be of different species. For that we must be familiar with the what that is said to be the artificial fertilization. So what is the process of artificial fertilization? In artificial fertilization, first of all, we must ensure that the pollen grains of the flower, hermaphrodite flower, should not be transferred to the stigma of that flower. So the pollen grains chosen should be removed from that plant. That process is said to be the emasculation, in which the pollen pocket is removed from the flower. Next one is the bagging, to avoid, so that not other kind of pollen grains should not fall on the stigma. So we bag, we means we keep some kind of polythene bag above the surface of, or the above the stigma of the female part, so that other kind of pollen grains should not fall on the stigma. Next one is the tagging. Then you mark the date of emasculation, bagging and transfer of pollen grains on which you have done the cross pollination. So you have done the emasculation, you have to do the emasculation to ensure, so that the pollen grains of your interest should be capable of undergoing fertilization with the stigma. Bagging is to avoid other kind of activities like contamination of some other kind of pollen grains, and tagging is to ensure that what you have done till date. So this is all about the process of outbreeding, pollen pesticides interactions, and artificial fertilization and the double fertilization. So in this we are capable of knowing how can we ensure self breeding, how can we do artificial breeding, and how the pollen pesticide interaction help us to study the process of fertilization. That's it. Thank you.